So here's the PowerPoint. We're going to start off right with chapter 1. This is the main themes of microbiology. Um, um, and this covers a variety of things. It's sort of a catch-all chapter. They talk a little bit about who the microbes are, uh, what sort of things do we study, what are the organisms and the organism-like forms that microbiologists are expert in or interested in. Discusses history, which is important because if you don't understand what's already been done, you don't understand what has been done and has failed, you're liable to try to repeat that, and you'd be trying to reinvent the wheel, so to speak, when in fact there's no point in that. So to know what questions are still burning questions that need to be answered, have yet to be dealt with fully, those kinds of things. That's where some grounding in history is important. And there'll be a few other things that we'll touch on as we go through this chapter. So let's go ahead and get into this. All right, so what is microbiology? Well, it's a relatively youthful branch of biology. It only goes back 250, 300 years. And I'll let you think about why that is. Older, other branches of biology, like anatomy, for example, are relatively ancient. One could argue that ecology, the study of interactions of populations in natural settings, that goes back thousands of years. But microbiology is something that didn't really exist prior to around the late 1600s, early 1700s. But of course now is incredibly important. And one branch of microbiology is the study of viruses, or virology. Of course, now we're in the midst of a pandemic involving a novel virus that no one had ever seen before. And that just points up even more forcefully how important microbiology is. So microbiology is the study of very tiny organisms and organism-like forms. Here they're referred to as acellular entities. Things like viral particles are not living. Prions, we'll talk about, are not living, and yet they reside in living things, and they have some features that are similar to living things and can cause disease, and so are an important part of this whole spectrum of things that we study. So those very tiny organisms and organism-like things are often referred to as microorganisms, or, well, here, I was going to write this word, but I only need to circle it, or highlight it. Microbes, right? Called microbes. And a lot of people hear that term and they have sort of a very kind of vague idea about what it means, but that's what we're referring to here, are bacteria and fungal organisms that are microbial, that are tiny, and protistans, which are more closely related to humans, animals, plants, and so forth than they are to bacteria. The archaea, that's another group that's bacteria-like. And then, finally, we have some very tiny animals, little worms, and even some, that are arthropods, like little mites and so on. Those are microbial. And then of course, you've got the viruses and the prions. We'll go through all of those later. In turn, we'll talk about each of them. But yeah, anything that's an organism or organism-like that's very tiny that requires the aid of a microscope to be able to see it is a microbe. And most microbes, it says here measure between 10 and 100 micrometers. Actually, that's kind of an overestimate of the size. I would argue that there are many of them that are very much smaller than that. If you look over here in this little, I guess, linear representation of the different microbes based on size, these forms in here, I should have bracketed those to begin with. Those are the viruses, and you can see their sizes are given with units of nms, which stands for nanometer. All right, that's a nanometer. And a nanometer, it says up here, well, here it is, this little metric chart. A nanometer is 10 to the 9th meters, which, if you put that in decimal notation, that's 0.00000000001 meters, 1/1000th of a meter, all right? And so you can see viruses are measurable in nanometers. That's an appropriate unit of measurement for those. And then we have the bacteria, which in this little thing here range from the low end, where there are the rickettsias, measuring about 1 micrometer, on up to the spirochetes, which are very slender. Their widths are much less than 1 micrometer, but their lengths in total measure about 10 micrometers. So we've got a lot of microbes in that range, a lot of very important microbes in that size range, measurable in terms of micrometers. So what's a micrometer? Well, if we go over here to our little chart, we see that micrometers, and there's that little weird sort of looking U at the beginning in the symbolism for micrometer. That's the Greek letter mu, mu. It's the lowercase u. That signifies micrometers. And that is 10 to the minus 6 meters, or 1/1,000,000th of a meter. Very tiny. If you took a meter stick and you cut it into a million little tiny slivers, each one would measure 1/1,000,000th of a meter. And that's about, that range in there, 1 to 10 micrometers is typical for bacteria and bacteria-like organisms. There's one group called the Archaea that we'll talk about that are closely allied with them. And then you can see in this range here, from 10 on up to around 100 micrometers in there, we have organisms represented in this little picture by the protozoa, which would include things like amoeba, trypanosomes, euglena, paramecium. All of those are protistan organisms, and they have much more complicated cellular structure than bacteria, and are quite a bit larger. They're metabolically very active cells. And if you were to look at one of your own cells, say, white blood cells, a neutrophil, something such as that, and compare the structure of one of your cells to one of these forms, say in the protozoa, you'd see a lot of similarities. And that's because they're all descended from a common ancestor. Animals, fungi, these algae, and the protozoa are all descended from a common ancestor and belong to one common group. So larger cells with nuclei and organelles and all those kinds of things are going to be in that size range from about 10 micrometers on up to about 100 micrometers. And then up here we have some things that are really large, like a flea. A flea is not a microbe because you can see that with the naked eye. If you want to make out fine details of it, you could use magnifying lens or something like that, let you know of their existence without using a microscope. You see one on your dog or jumping on you. And so they're outside of the range of things that we would be interested in as microbiologists, unless we wanted to study the living things that are inside of them, which, of course, there are many living in their guts and so on. But roundworms here, um, these are cysts of a roundworm called trichina living in some flesh, I think some pig, some pork, and they're larval forms that are kind of biding their time to eventually emerge from there and make their way out of the animal. Their size ranges on the order of a millimeter, and that's pretty big, but it's still small enough that you would need a microscope. So that's kind of the upper end of the size range of things. And then this is a fungus sporangium. It's part of a whole fungal organism that consists of several cells. So in these images here, we're talking about multicellular forms, and there are some of those that are microbial. All right, so it says most microbes are in that range, 10 to 100 micrometers. Know that there are some that lie below and above that range as well, quite a few of them. The upshot is we're talking about very tiny things. So there's that. So why study microbes? Why study microbiology? Well, I think, intuitively you understand it's an incredibly important area of study, but we'll help to kind of add some more specific details to your understanding of that, how to answer that question. So one reason is that if we were to look at all of the organisms on the planet and assemble them into different groups and then count the numbers of members of each of those groups, we'd find that the microorganisms are by far the most diverse organisms out there. That means there's more kinds of them than there are in other groups, such as animals and fungi and so forth. So we know several thousands of, say, the bacteria, but it's almost certain that there are many orders of magnitude more forms out there that we've not yet ever seen, living in out-of-the-way places that are difficult to get to. So that would tremendously inflate the number of kinds of things that we would list there for that particular group of microbes. And viruses are even more diverse. And as we're seeing now, we've got a new coronavirus that had heretofore not been known. And the thing with viruses, bacteria, all of these microbes, protistans, and even fungi, is that they evolve very rapidly. So there's lots of speciation. That adds to their diversity. They have very short generation times. That is, the time that it takes for one of these guys to come into existence until it then gives rise to others of its kind through reproduction, through replication. In the case of viruses, the time that it takes for that to happen is often measurable in terms of minutes to hours. And so populations of these things can turn over very rapidly. And if there's genetic variation that arises, if mutations and things occur, you can have change in these organisms, evolution in these organisms happening, in days. So new species can arise very rapidly. So they're incredibly diverse. And then, in terms of numbers of individuals within populations. So if you take a population of E. coli, say in the gut of a human, the number of individuals there is enormous. It's trillions of cells there. And in terms of mass, they wouldn't weigh very much. But if you could actually see them under a microscope and count them, you would be overwhelmed. So you have in your own body, just one human, you have more microbes living on and in you than you have your own cells. And within your own cells, you have organelles, including mitochondria. And it turns out, we'll talk about this later as well, mitochondria are actually obligately living bacteria that are found in your cells. They evolved to the point where they lived in cells and have taken up that particular lifestyle, and that's all they do. Mitochondria don't live freely in the environment anymore. Billions of years ago, they did. And so, if you were to count all of your cells, you have about 37 trillion or so cells. In each of those active cells, you're going to have several hundred mitochondria. You're going to have 37 times 10 to the 11th power or so mitochondria. That's no, to the 14th power, excuse me. 100 trillion or more of those in your body. So that's enormous. We're talking huge numbers that most of us can't even fathom. So that's just in your body. You think about all the billions of people on the planet, and think about all of the other organisms that are carrying these microbes, and all the other settings outside of bodies of living things where they can live. I mean, it begins to set in that the world as we know it is dominated by the microbes. They are everywhere, and their activities are essential to the planet, and life on this planet. One of the ways that they're important is that they help to recycle essential elements. So as organisms live and they metabolize and they take in materials and incorporate them into their bodies, they reside there for the duration of that organism's life. But when that animal or that plant or that fungus or whatever it is dies, it goes through a process of decomposition. Its body breaks down. Now, that's not a given that will happen, except in a world like ours where we have microbes. And so what causes decomposition? It's the action of these microbes, bacteria and fungi acting on bodies, literally tearing them down, breaking them down. And as those bodies sort of, dissolve into the soil, their elements are released. So the various kinds of important elements like carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, and other important molecules, amino acids, simple sugars, all of those things are now available. Whereas if that organism had died and had not decomposed, its body would have retained those things. And ultimately, as living things, began to multiply and die, if they didn't decompose, all of the available elements for life would be locked up in dead bodies, and life as we know it would cease. Right? So these elements are merely borrowed for a period of time while the organism is living, and then they're allowed back into the environment so that they can cycle yet again and life can arise anew. So that's cool. And microbes very surely are pivotal in that process. They are the keystones of that process. Microbes themselves are sources of nutrients. So for some organisms, they're important items to be consumed. We have a number of organisms that live on microbes that they feed on. And in fact, you know, many organisms, many microbes provide nutrients without having really to be ingested purposely. You have in your gut right now trillions of E. coli. Um, Escherichia coli is a species of bacterium, and those active E. coli produce things like vitamin B12 and vitamin K, and thereby provide some elements that can help you out, that can help your own metabolism. So we've got microbes doing that sort of thing, providing nutrients in various ways. And then, we think about this photosynthesis as being the main sort of occupation of plants, trees, and low-growing forbs and grasses and so on that have leaves that are green, right? You know, the world outside is green, and all of that green stuff out there, those plants have cells with chloroplasts, and in those chloroplasts, we have structures that contain chlorophyll, which is a molecule that absorbs sunlight. Well, as a result of that, that energy can be put to use to help make food. So photosynthetic organisms are taking carbon dioxide, which is inorganic and doesn't have much energy on its own, and they're taking water, which again is inorganic and doesn't have much energy on its own. You know, it's not like you drink water to get energy from it, but it's helpful in other ways. But what these organisms do is they take carbon dioxide and water and they recombine some of their parts. And dog bugging me here. They recombine some of these parts, and that arrow, and they produce glucose, C6H12O6, and oxygen, molecules of oxygen. That's supposed to be a 2, O2. Awful, sorry about that. All right. Anyway, that's what photosynthesis is. Taking carbon dioxide and water, I apologize, I blanked that out somehow. Carbon dioxide and water and recombining them to make glucose, C6H12O6. And in this C6H12O6, the sugar, you have carbons, and bound to those carbons are lots of hydrogens. And the bonds between those, that little bond right in there, that single covalent bond, contains some energy. And so when you eat sugar or ingest some other carbohydrate that has carbon and hydrogen bound together, your cells are equipped with enzymes that will split those bonds and release energy. And so that's why we eat food, is so that we can get those molecules to do that for us. So photosynthetic organisms are making food on their own using light. And a lot of the microbes are photosynthetic. So, like I said earlier, it's the plants that we generally think of as being the kings here with regard to photosynthesis and the extent of it. But the truth of the matter is there are lots of single-celled, very small multicellular organisms that are photosynthetic as well, and they're microbial. We'll talk about those later, but know that that's an important thing there. So, and then we've got all these other things that we have learned to get from microbes. We've learned how to exploit them in these ways. We've figured out ways to grow them on purpose so that they can make these things for us. So lots of food items are made with the help of organisms that ferment substances in those food items. Um, so if you've ever had kimchi, which is a Korean um, sort of staple, it's made with cabbage that's been shredded and some spices have been added to it, and they allow naturally occurring bacteria to act on it to ferment it. And people enjoy the taste, and actually it's more nutritious than eating just the cabbage by itself. Of course, there's sauerkraut, um, which has been made for centuries, and real sauerkraut is made by fermenting shredded cabbage. And then you have beverages, like beer and ale and mead and various things like that. The alcohol content of which is a result of bacterial metabolism, fermentation of sugars. Antibiotics, penicillin, the cephalosporins, erythromycin, all of those different kinds of antibiotics are in fact products of bacterial metabolism. A lot of people don't realize that, but they depend on them for that. And then many vitamins, the production of vitamins and all kinds of things, acids, a variety of things that are valuable to us in many ways are products of these organisms. So they're hugely important there. Most of what I've been saying about these guys thus far, you could say is pretty positive. They are important in beneficial ways. But there is this, of course, downside we're experiencing now with the virus that's basically still raging in the world. We've got viruses, and we've got some bacteria, and we've got some protistans that cause disease. And the word for organisms that are disease-causing is pathogens. They are pathogens. So a number of them are. Now, interestingly, if you were to look at all of the bacteria and determine which of them are strict pathogens that will always cause disease, the number of forms is relatively low. So most forms are not simply, designed essentially to be pathogenic, to be disease-causing. There's a few of them. The measles virus, for example, it will cause disease every time, and is one that could be regarded as a strict pathogen. A bacterium like Bacillus anthracis that causes anthrax, one could argue that it is a strict pathogen, and there's a few of them like that. Most of the others that cause disease only do so opportunistically, so they'll cause disease only when conditions are right for it. When they occur in a normally healthy person whose immune system has not been compromised, there are no comorbidities as we're seeing these days, they'll often live there without causing any harm at all. They might be neutral, they might not be helping, but they're certainly not hurting. There are lots and lots of organisms right now in your body that are going to fit that category. But as far as numbers of organisms that strictly cause disease, there's not a tremendous number of them among these microbes. But it is an important area. Most students in this class, I suspect, are interested in these clinical aspects of microbes, so we will talk about that some. But know that there's this whole other base of things where disease is not something that even enters into the equation. So a big question has to do with where the microbes came from. I've been hearing discussions about viruses and their evolution. Where the heck did these viruses come from? Why are they here? Why do we have to deal with them? Questions like that are very significant in biology. We have people who study um, uh, evolution from this perspective, and what they've determined is that uh, the Earth is 4.5 billion years old. Origin Earth is about 4.5 billion years. It does that. BYA means billion years ago. So 4.5 billion years ago, the Earth arose, okay? Likely through the consolidation of cosmic debris following a Big Bang event, which occurred 15 billion years ago. Um, and there's ample evidence for both of those things that are beyond the scope of this course. You'd have to take a course in um, geology, paleontology. I think they might, or uh, astrophysics, talking about cosmic evolution. But yeah, 4.5 billion years ago is the generally agreed upon origin of the Earth. And then some billion years or so ago later, actually it's closer to 3.7 billion years ago. So a little more than 3.5 billion years ago, as it says. We have evidence available in rocks here on Earth now that there were living things at that time. Prior to that, older rocks don't seem to have any evidence for that. It stands to reason that that would be the case. So it took around 1 billion years for the first very, very simple living things to arise. And then from there, obviously, there was this diversification of things to get us to where we are today. And those first living things, um, the few that we have embedded as fossils in rock, appear to be most closely similar to forms we have today that are referred to as prokaryotes. So very simple forms. And those prokaryotes are of two kinds, and we'll talk about them more later, but generally just know that these are the bacteria. And another group whose name means ancient are the archaea. Closely similar, different enough though that they are separated out into separate categories. Very simple, very small cells. These are the closest things that we have with us still today that could be considered like those forms that arose about 3.5, 3.7 billion years. Of course, later, more complex organisms arose. Those are the eukaryotes, and that's the group to which we belong, and all of the other so-called higher organisms that have complicated larger cells. All right. And those arose, as you can see down here in this little timeline. There's the eukaryotic cells. If you follow this down, sometime around a couple of billion years ago. Some people actually push this back to right around in here and make the claim that the eukaryotes arose about 2.5 billion years ago. But certainly, their origins are much more recent than those of the first living things, the first prokaryotic cells, running back to right in here, 3.7 billion years. All right, so the upshot is the Earth is ancient. It took a long time for living things to finally evolve. They started off as very simple self-replicating molecules and then gradually became more complex from there. They had, of course, the evolution of nucleic acids like RNA and DNA and cellular structure. And, things went on fairly simply for good long time, about 3.7 billion to 2 or so billion years ago. And then from there, you can see in this end of the timeline, this burgeoning of life on Earth. We had a massive sort of explosion of forms beginning, actually right around in here. So about 500 million years ago, we had a big explosion of living things on the planet. Right? So the microbes are prokaryotes and some of the eukaryotes. Okay, not all eukaryotes, but some of the. And then this is a kind of a really streamlined tree of life. We could zoom in on it on any one of those branches and see that there are many, many, many smaller branches within them. But this just kind of generally, sort of grossly overviews the different groups of organisms on the Earth as we know it. And you can see each little cluster of organisms is sort of self-contained, and they all trace back to a single common ancestor. So here we've got all of these forms here that trace back to an origin, a root, about 0.6 billion years ago. The animals, right? All these multicellular organisms, most of which aren't microbial, but we've got some flatworms and nematodes that qualify. So we'll be talking about those later. The fungi make up a second group, some of which likewise are also microbial. Some of the molds, for example, and molds can be in each of these three major groups that are shown up here. And there's other groups of fungi, by the way, besides Ascomycetes and the Zygomycetes and Basidiomycetes. But anyway, suffice to say, there are some microbes in that kingdom. And then the plants are here. They're a group off to themselves. Now the plants, I'll put a little asterisk by them. They get an asterisk because they're the oddballs. That's the only kingdom that doesn't contain any microbes. So while they're super important and microbes live on and in them, they themselves do not contain any microbial forms. This kingdom doesn't. I'll put an X through that to indicate that we can leave them out of the discussion for the most part. Okay, so the fungi contain some microbes. The animals contain some microbes. And then we've got down here, you can see this big old mass of things across the base of this tree that's growing has branches that lead off to the animals, and a branch that leads off to the fungi, and a branch that leads off to the plants. They're very closely related to all of those different forms. But these protists constitute a kingdom unto themselves, right? And you can see they're incredibly variable. It's considered sort of a catch-all group. If something doesn't very clearly fit into the plant kingdom, say, taxonomists have been prone to in the past just throwing them into this kingdom. If something is fungus-like but not really a fungus, let's put it in the Protistan group, and likewise for the animals. So sort of a catch-all group. And many of these, the thing about these protists, many of them are microbes. Lots and lots of them are single-celled. Lots of them are important as decomposers and disease-causing organisms, and in a variety of other ways. So we'll be talking about those. And so the protists are largely, not entirely microbial. There, for example, are some of these algal organisms here, the red algae, the brown algae, and the green algae. A lot of those are very big organisms, be considered multicellular, but they're still listed as protistans. All right, so this whole top of the tree here, the whole crown of the tree, that constitutes one group of organisms known as the eukarya. All right? So plants, fungi, animals, and protistans are eukarya. or as we call them, eukaryotes. Right? They're the eukaryotes. And if you're familiar with taxonomic hierarchy, the different categories that we place organisms in. We start off with the species, and we go to the genus level, and we go to the family level, and on up through the different levels you can place organisms in. It's species, genus, family, order, class, phylum, kingdom. Now we have a level above the kingdom level, and it's called the domain. So the eukarya is actually the name for a domain of living things. So you're in the domain eukarya, as are all of these guys in this upper part of the tree, the plants, the fungi, the animals, and the protists. And then way down here at the very base or the root of the tree, you can see coming off early on in the evolution of life on Earth, we have two groups. We've got the archaea and the bacteria. And some people formerly called them monarans, and there was a kingdom monera. We no longer use that. So that is an antiquated thing. Just disregard that entirely. Now what we say is that the bacteria are a group unto themselves. They constitute a kingdom and a domain, right? That's kind of weird, but yeah. So they're a separate kingdom and domain unto themselves. And likewise for the archaea, right? That's another domain / kingdom of living things. So these are domains. I'll put D and K here to indicate that that's a domain and a kingdom. Then a D and a K here to denote that archaea constitute a separate domain and kingdom as well. And it says they're odd bacteria. I should have revised this. I borrowed this slide from someone else and just now realized that calling them bacteria is actually, inaccurate. They are not bacteria. They are a group unto themselves. At one time, they were considered to be very odd, unusual bacteria that took up a lifestyle that most other bacteria couldn't handle. But now we know through the efforts of a variety of important biologists, the most important of which was a guy by the name of Carl Woese at the University of Illinois. Back in the 70s was when he did most of his work. Through his efforts, studying the molecular biology and other aspects of these things, we've now elevated these archaea to a status where they are independent of others. All right, so the archaea constitute another domain and kingdom, and they are microbial. So the bacteria are microbial. Every last one of them would be. The archaea are microbial. Every last one of them would be. And the eukarya contains some microbes. The protistans, largely microbial. The animals, contain a few microbes. Fungi contain a large number of microbes. Plants don't contain any microbes, so they're not part of our discussion. So those are the living things that are microbial, excuse me, that exist in these three domains of life. The eukarya, the archaea, and the bacteria. All right, and this just goes a little bit further in comparing the domains, bacteria and archaea, kind of pointing up, emphasizing how it is they're different. Some of the things in these lists of attributes are a little redundant, and oddly placed. So bacteria are single-celled. Well, so are the archaea. But they're not different in that regard. But this is one major difference. The bacteria contain a substance we'll talk about extensively later called peptidoglycan. There's a whole chapter on just the prokaryotes, and we'll see that peptidoglycan is really figurative in that discussion. Peptidoglycan is unique to the bacteria, so archaea don't have that. All right, so that's an important thing to know. Most of the bacteria, in fact, all of them lack a membrane-bound nucleus. There are a few that might have some of the cell membrane invaginated a little bit to surround the genetic material, but not a full-fledged nucleus. So, in fact, that the name prokaryote, which bacteria and archaea are considered to be prokaryotic, literally means primitive nucleus, and in fact, they don't really have a true nucleus. So there's that. This word here is important, ubiquitous. It's a good one to add to your vocabulary. You can throw that around and show how smart you are at barbecues and things like that. But ubiquitous means everywhere. So if something's ubiquitous, it occurs everywhere. So bacteria are literally everywhere on the planet. There are some places that have a whole lot more than others, but just about everywhere you go, even to the most inhospitable, extreme places, around volcanic vents and down in the deep ocean and elsewhere, you'll find some bacteria there that are scrounging out a living somehow or other. Bacteria are also the main members of the microfloras of larger organisms. So we've got a microflora in our gut. That would include our mouth all the way down into the small and large intestines and rectum, and then on our skins and up our noses, and in some other places we in fact have these bacteria. And there's a few archaea there. You might have some fungi. You may even have some protistans, but the vast majority of these are bacteria. And you might be thinking, well, that's awful, that's gross. These things are exploiting us and taking advantage of us. But in fact, the truth is our bodies wouldn't function optimally without them. So it's almost like we have another organ system, and we're learning more and more every day about just how important that is. So anyway, we'll talk about that some later. But just know that when we talk about this microflora, we're talking about microbes that live on and in bodies of larger organisms, and bacteria are probably the most important of all of those. And then the archaea, this other group, one thing that sort of made scientists aware that they were unique is the fact that at the molecular level, specifically the genes in their genomes that code for rRNA, which is the main component of ribosomes, those genes, when they sequenced them, that is, they determined the arrangement of adenines, cytosines, thymines, and guanines in them, and they compared those sequences with known bacteria, they saw that, hey, these guys have to have separate, independent evolutionary histories for much of the time that they've existed. So it constitutes a group that's split off entirely from the bacteria, and now, deserves this recognition as a separate domain. Here's another important point. There is no peptidoglycan in the archaea. Another sort of plank in the platform of the people who really pushed for designating them as a separate domain. So, no, they don't have peptidoglycan. They have other substances, some of which are unique to the archaea, and we'll be talking about that as well. Those unique membrane lipids. And it mentions here that some of them are methanogens. They can produce methane. So a lot of methane, CH4, is natural gas. and deposits of that occur in old places that formerly had been swamps and forests and so forth. Um, we can tap into those places, drill in, and extract that natural gas. Well, a lot of it was produced by the archaea, acting on ancient uh, plants and things that had been buried and covered by soil for millennia. And the result of the metabolism of that material is the production of CH4. And archaea can do that. And then another thing about the archaea it's not unique to them. There are some bacteria that can do this, but they probably are much better overall than bacteria and everybody else at finding and living in extreme places. So many of these archaea are poorly known for that very reason. It's hard to get to the places where they live. So some of them live deep in the earth. If you have some porous rock, for example, into which you can have seepage of water and things, some organisms find their way down into the depths of the rock. They're that small, and they can live there, making use of very paltry supply of nutrients and things. As I said earlier, they can live around volcanic vents, live in the deep ocean. They can live in places that are highly acidic or highly alkaline. They can live in really salty places. So places that are inhospitable to other organisms, the archaea tend to thrive there. And that's probably why they occur there. They don't have to compete with anybody else. So anyway, a little bit about those two domains of living things that we'll be talking about much more extensively later. And this little section here just kind of whips through some of the other forms that we've talked about. So the eukarya, the domain eukarya, the protists constitute a kingdom. Okay, so for now, that's a kingdom in the domain eukarya. I say for now because there are some people who are really pushing to take the protistan group and break it up into a bunch of additional kingdoms. So they think that it deserves to be revised. And those changes are on the way. The good news for you is that they've not been implemented fully yet. So if you were taking this class, say 20 years from now, you might have to learn like 20 or 30 new kingdoms. But for now, we're going to talk about this as one big kingdom. And so, yeah, some of them are plant-like. These are the photosynthetic algae. They're protists. Others are more animal-like. They can't photosynthesize, but they live by feeding on other organisms, so we call those protozoa. And both of these are not official names. So there's no class algae or phylum algae or likewise for protozoa. Those are just sort of general terms for the organisms that fall under those headings for. And then we have slime molds and water molds, which defy pigeonholing into anyone. They have attributes of fungi, but then they have attributes of other kinds of organisms, so they are considered to be protists. And down here you can see a couple of pictures. These are some slime molds here. This yellow sort of looks like some yellow slimy material kind of bubbling up out of the mulch here in somebody's flower bed. Those, in fact, would be slime molds preparing to kind of coalesce in one big mass and reproduce and carry on with their life cycle. You can see that sometimes. And then this potato, you might look at it and say, oh, it's just rotten. It is, in fact, being decomposed, but not by some bacterium or fungus. It's actually a type of water mold that's acting on it. And the significance of that is that in the past, populations that were really dependent on potatoes, like the Irish in the 1800s, suffered mightily as a result of the spread of water molds that devastated their potato crops. So somewhere around, say, I've seen different estimates, but around 5 million people died in Ireland in the 1840s as a result of failure of the potato crop. That's how important potatoes were for them. And it was a protestin. It was a water mold that caused it. Now we have water mold resistant varieties of potatoes, but even today it still happens. And then the eukarya also include the kingdom fungi, so I'll put a K there to indicate that's a kingdom. I pronounce it fungi. Some people pronounce it fungi. Either way is correct. But without belaboring it, let's just say that none of these are photosynthetic. They all have to make a living by absorbing nutrition from the surroundings. They're important decomposers. They do a number of other jobs as well. We'll talk about it. I don't want to let the cat out of the bag too early, but yeasts, there's a bunch of different yeasts. This particular yeast that's shown here is called Saccharomyces, and there's a number of different ones of those. But that name literally means sugar yeast, or sugar fungus, because, as you know, if you were to take some dry, inactive yeast, mix it with some water to get it metabolically active, and then add a little bit of sugar, it'll begin to break that sugar down to produce carbon dioxide, release bubbles of CO2, which, if we were to add that to some dough, as the yeast acts on the sugars there, it'll produce bubbles of CO2 that get trapped in the dough, and it makes bread, have these little air holes all through it. Makes it palatable and more desirable to eat. But ultimately, if the bread sits too long, other fungi can take up residence in it, and you'll see mold. So in molds, a variety of them are also fungi. So bunches of different ones of these. Some of them are massive multicellular forms that you can see obviously with the naked eye. But much of the life cycle of many of those species even would be microbial. So we have important microbes in this group. All right, and so this just shows how through time, this is a much more detailed picture of the tree of living things than what we saw previously. And it contains some information in it that has only been brought to light in the last 20, 30 years. But you can see here's our ancestor. We don't know who that is, but that's the single living organism that arose about 3.7 billion years ago, from which we had all of these lineages arising as that population diversified. And you can see you've got lines that run directly to particular destinations, like the chlamydias and the spirochetes. That lineage is a lineage unto itself. And the same is true for the gram-positive bacteria and the endospore producers, which are positives as well. But then you see here among the gram-negatives, right, we got this line that runs up to them. But then, weirdly, there's this side branch that comes over. Look where it goes. It goes to the eukarya. And then that same thing happens. We come up here and going to the cyanobacteria. Hey, we got a side branch. Boom, it goes over here to the eukarya. So what happened? Well, it turns out that in both cases, some members of these two groups, the gram-negative bacterial clade or group and the cyanobacteria, took up residence in cells of eukaryotes and became important organelles. So what they're showing here, what they're trying to depict, this would be a line that corresponds to the appearance of mitochondria in the eukaryotes. So At one time, mitochondria were um, free living gram-negative bacteria, but they came to take up residence in a way that we'll talk about later. We'll discuss that, the proposed hypothesis about how this happened. They took up residence in eukaryotic cells and became mitochondria. All right? So mitochondria in your cells really are, at their deepest level, they are bacteria. They just happen to live in your cells now. That's the only place we find them, is in eukaryotic cells. And then this line here that's going from the cyanobacteria over to the eukaryotes, that represents another kind of organelle that's found only in photosynthetic organisms, photosynthetic eukaryotes, the plants and others. They're the chloroplasts, right? So you might think there's no way any of these branches can, any one limb is going to have branches coming off of it, but in fact that's happened. So yeah, these are all the living things. So the eukarya make up a domain unto themselves. The archaea make up a domain unto themselves. And the bacteria make up a domain, although some of their members have come over and joined forces with eukaryotes as mitochondria and chloroplasts. Okay, so those are the living microbes, right? But not all microbes are living. Not all microbes are cellular. And then this thing here, you can look at this on your own. This shows representatives of different groups of microbes. We got the bacteria here, the fungi. We got some single-celled and short chains of cells of algae. These are all protists. Here is another protist, one that's not photosynthetic, that has little cilia on it. So this is a free-living protist that lives in swampy places. And then here's some little encysted worms in some flesh of a hog, or sometimes bears have these, or even raccoons. Those are microbial animals. This picture here shows some viruses. Right? Those are viral particles. And you can see for all of these, they've given the extent to which those images have been magnified through a microscope. You can see there are pretty high magnifications, but look at this. For the viruses, that image has been magnified 100,000 times. 100,000 times. That's crazy. So that was taken using an electron microscope. The upshot of that is viruses are really, really, really tiny. And anyway, they are microbial, but they're not cellular, right? So we need to add them to our list of things that we would study in microbiology, because they have some features that are like living things while departing in other ways. And so here, it's just a little bit on those forms, these acellular microbes, which are in all cases infectious. The thing about viruses and these other forms, they can't exist without taking up residence in living organisms. They have to go to those organisms and live in their cells and use the cellular parts. They say they hijack the cells and they take them over and use their parts for their own purposes. And this is why you get sick when you are virally infected. The virus is taken over your cells and is disrupting their function, even killing many of those cells in the end. So here's a term that describes both viruses and this group here, the prions. They are obligate intracellular parasites. So we can say that same thing that applies to these as well. In fact, all of these things that are acellular, non-living microbes are this way. They wouldn't exist otherwise. Obligate means they have to. They have to do it. It's necessary. Intracellular means they have to go into a cell. within a cell. Parasite means they take and they benefit while the host that they're living in is harmed. Parasitism is a plus-minus relationship. The parasite wins, the host, which would be you if you got a virus, loses. Right? And viruses are pretty simple. They are in no way, similar to cells. Not really. They have always a little bit of genetic material. It's either DNA or RNA that they use, and we'll be talking about that. There's a lot of variation in their nucleic acid structures, but suffice to say they have some genetic material. And then the rest of their composition is mainly protein, although some of them, like this HIV virus, has on its outside an envelope, which is derived from the host cell's own cell membrane. So they'll often have an envelope. The coronavirus that we're dealing with now, the SARS coronavirus number 2, remember there was a SARS coronavirus number 1 several years ago, and that was eventually contained and did not cause any further problems, unlike what we're dealing with now. These coronaviruses and influenza viruses and viruses like HIV all have this additional structure outside of themselves, called an envelope, and it's derived from cell membrane. But mostly, that's what they're made of, is nucleic acid and protein. And they cause a variety of diseases. There can be asymptomatic infections as well. And then many cancers are now known to have viral origins. Sometimes that happens. You'll have virally infected cells whose DNAs are disrupted and they will become cancerous. And then we have prions, and prions are weird. Unlike viruses, they are strictly protein. That name prion means, or is derived from the term proteinaceous. I apologize for the poor writing. I'm doing this with my finger. Proteinaceous infectious particle. They're proteinaceous infectious particles. And that's all they are. They're proteins that somehow enter the body, and they take up residence preferentially in the nervous system, central nervous system in particular, where they do damage to important parts of the brain, and ultimately lead then to cognitive and balance issues and so on, and ultimately leads to death. There's no recovering from a prion infection. It says here they're composed of strong tertiary structures. You know how proteins have different structures. They start off as primary structures, just a chain of amino acids, and then that gets either twisted into a secondary structure, a helix, like a coil, or a pleated sheet. And then finally, the tertiary structure is just a glob. It's where you got a coiled protein that is rolled into a big wad, or a sheet-like protein that's rolled into a big wad, like taking a piece of paper and crumpling it. That's how these proteins are shaped. They're globular. And it says they're strong tertiary structures. What that means is they can't be broken down as readily as other proteins would be. So normal proteins in your cells can be denatured by a variety of things like heat and acid. Weirdly, the prions resist that. So prions are heat resistant. They are resistant to a number of disinfectants. Imagine the strongest, best disinfectant you can think of. It won't have any effect on a prion. Heat, like you would have, say, with a pressure cooker or through boiling, won't break down a prion, right? So when it says strong, that's kind of an understatement. They are some of the most resistant forms of microbes that we have, which is really scary because, autoclaving and pressure cooking and those kinds of things in many cases won't even disrupt them. A couple of diseases that are attributable to prions. One is called BSE. That stands for brain fungiform encephalopathy. I won't try to write that with my finger, but the common name for that is mad cow disease. So cattle can be infected by prions that will cause them to be sickened, and they will die ultimately. Another disease that humans are affected by is called Creutzfeldt-Jakob disease, which is a mouthful and a lot to write down. So most people call it CJD. What you see in this picture over here, brain of a person who has been stricken by CJD. You can see it looks like it's shriveled. It looks like it's just kind of collapsing in on itself. It's nowhere near as plump and healthy looking as the control brain over here. And that signifies destruction of brain tissue. So that brain ultimately is not going to be able to function to do the kinds of things that people need to be able to do. And another prion disease now that is circulating in our wild deer populations is known as CWD, chronic wasting disease. And it preferentially attacks deer. So that would be white-tailed deer and mule deer that occur out west. And moose, those are also deer. So there's some prion that's sort of specific for those kinds of animals that causes a similar set of symptoms. And we don't know with either BSE or CWE how likely those prions are to cross over into human populations and cause infection there. But that's concerning, and a lot of people would say that to err on the side of caution, and if you have some means of testing for CWD or BSE in meat from, say, cattle or from deer, to utilize that and avoid eating meat contaminated. So anyway, viruses and prions are non-living, okay? But we do consider them to be microbial. And then here's an addition to the list of things I said earlier about why microbes are important. We could go on for a long time discussing these things. Let's just go through a few of these. I've already mentioned decomposition, about that. Here's another one, nitrogen fixation. So nitrogen that occurs in the atmosphere occurs in molecular form. Two nitrogen atoms joined together. And actually the way that molecule looks, if you could actually see it structurally, it's two nitrogen atoms joined together with three covalent bonds. Now, the importance of that is it's very challenging to work with that nitrogen that's so tightly bound together, be able to add it to other molecules. And it turns out that a lot of organic molecules that are important in living things require some nitrogen. And nitrogen is the most abundant element in our atmosphere. It's hugely available. There's no limitation on nitrogen's availability. It makes up 75% or more of the atmosphere around us. But because of this structure, because of its resistance to entering into chemical reactions, it is in essence just almost inert, which means unavailable to most living things. So you can't breathe in nitrogen and incorporate that into molecules in your cells, like proteins and nucleic acids. It'd be nice if you could do that, but you can't. And, just know this, we're utterly dependent on that nitrogen. We need it. So how do we get it? There's a paradox. It's something we need. It's highly available. We can't get it on our own. We're not equipped with the enzymes to do that. Well, it turns out many microbes are. And so here's the name of the process whereby they take up that nitrogen and incorporate it into molecules that we then can use. All right. What they'll do is they'll take the nitrogen out of the air and they'll take N2 and then make some subtle changes to it. One of the molecules that can be made with it is ammonium, and another one would be nitrate. I believe those are the symbols for those. So these nitrogen-fixing bacteria can do this. Now, when the ammonium or the nitrate go into the environment, say into the soil, plants can take these substances up, right, and incorporate them into their molecules for making proteins and nucleic acids. And then we, in turn, will eat the plants or eat animals that ate the plants and pick up our nitrogen that way in the form of amino acids. And so getting nitrogen into a cycle where it passes from one living thing to the next begins with this process of nitrogen fixation. So you better bet that's incredibly important. Life as we know it on this planet wouldn't be possible without that. Now, do all microbes do this? No, it's only a select few. There's a couple of different genera of bacteria that are capable of nitrogen fixation. I'll just give you the names of two of them. One is Rhizobium. So these are bacteria. Rhizobium. And another one is Azotobacter. There are some blue-green algae, which are microbial protozoans or protistans. They can do this as well. But not everybody can do it. It's just a few things have the genes that code for the enzymes that allow them to. That's what they're doing. They're performing nitrogen fixation and making the environment more hospitable for them. Interestingly, Rhizobium and Azotobacter often live in little tumor-like growths on the roots of members of the bean family. So beans like soybeans and others, or clover, which is in that family, or alfalfa, which is in that family. Locust trees like black and honey locusts, often their roots will have these little tumor-like growths. So you'll see the roots going through the soil. You'll see these little masses, little spongy masses forming on their branches. And in those is where you're going to find a lot of bacteria that are doing this nitrogen fixation. A lot of farmers actually plant those plants on purpose to nitrify the soil, to add nitrogen. They may not understand why that happens, but now you know that those plants harbor these bacteria, which in turn are nitrogen fixers. And of course, photosynthesis, that goes without saying. Here, organisms are doing is that they're taking carbon dioxide out of the air and putting that to good use. They're fixing the carbon, and so the sugars and the starches and all of those kinds of molecules that are products of photosynthetic organisms would not be available to us were it not for photosynthesis. That, of course, is a way that organisms are exploiting sunlight, and that drives everything. That's all important. Without that, life as we know it would be impossible. We have a number of microbes that enter into what are called symbioses. There's this fancy word, symbioses. A symbiosis, that's the plural of the word symbiosis. If I replace the E with an I, a symbiosis is just a relationship between two organisms that live very, very, very intimately, very closely with one another, such that if you were to take any one of those, any member of the pair out, the other one would fail. They would not do as well separately as they do together. And there are a bunch of cases of pairs of organisms that have evolved to have this kind of association. So mycorrhizal associations. Mycorrhizae, the word from which mycorrhizal has been derived, mycorrhizae, that word refers to certain fungal organisms that are microbial that attach themselves to the roots of plants. So they don't form little tumor-like nodules, but instead what they do is the fungal structure is like a thread. Right, the cells in the fungus are strung together in these thread-like, very thin filaments, and what they do is they attach to roots and they wrap around them and form this kind of cottony, soft coating around the root. And you might think, well, gosh, it's going to choke that root out. In fact, it doesn't. What it does is it makes the root work better. Roots are in the soil to absorb water and nutrients. Well, a root that's been associated with mycorrhizae, a root that has struck up that sort of symbiosis with a fungus, will be able to absorb many, many times more water and nutrients than would have been the case otherwise. So that's hugely important. So many of the trees that you see growing, if they're really thriving, if they're doing well, in whatever situation you find them in a forest or woodlot or in your yard, even smaller plants, they do best when they have these mycorrhizae associated with their roots. So yeah, you look at an organism and think, oh, you don't realize that often organisms that do well are doing so because they have helpers. And it would be other organisms, kind of like you. You've got E. coli in your gut. They're helping you, right? So no organism is an island. They all sort of interact. And of course, we've talked about already chloroplasts and mitochondria. We'll come back to that, but those are derived from bacteria. And then there's an extra S in here. Take that S out. So parasitism. Yeah, parasitism. That's disease. So unfortunately, that's a type of symbiosis too, but in this case, instead of a win-win, where the organisms are helping each other, the parasite is harming the host, and that is a symbiosis. They're living together, but not in a functional way. It's a dysfunctional thing. In the case of mycorrhizae and other kinds of symbioses that are helpful to both parties, that's an example of a mutualism. Not parasitism, but it's a mutualism. So I'll put plus plus to indicate that's a win-win for the two parties involved. In the case of parasitism, like if you were to be infected by, say, God forbid, Clostridium difficile, got really sick, that organism is happy because it's got a place to live and it's doing well there. You're, on the other hand, sick and maybe even find yourself in a life-threatening situation because of it. That is a parasitism, a plus minus. So the parasite's gaining, the host, even you, if you got sick, loses. So yeah, this just details some more about these activities that microbes are central to. So photosynthesis and decomposition, other uses of microbes, biotechnology. I've already mentioned the production of many things that are vital to our continued existence on the earth. So all kinds of things involve the use of microbes in ways that we had to engineer the ways that these things are used. Over here, they've got some tanks full of protist and algae in this picture. And those organisms are producing products that then can be extracted. Among those would be things like jet fuel. There are alcohols that some of these organisms actually produce that can be extracted and used. So some pretty extreme things going on there in many cases. Um, Genetic engineering, this is where you take DNA from one organism and manipulate it by recombining it in many cases with DNAs from other organisms. So you could take, say, a desirable piece of DNA from, I don't know, say, um, tomato, right? Take some DNA out of tomato cells and remove a gene from one of their chromosomes that code for something that would be desirable. There's a substance in tomatoes called lycopene that they can make. So maybe lycopene has some sort of therapeutic use. I know that it's helpful in restricting the growth of cancer cells in men who have prostate cancer. That's something that's been hypothesized. So let's say you wanted to get a whole bunch of lycopene without having to extract it from tomatoes, right? What you could do is take some of the genetic material from the tomato and splice it into another organism's genome, and that organism then could be the one that synthesizes the lycopene. And they do this a lot with bacteria. So you could take the genome of a bacterium, say E. coli, and add the tomato gene to it, and then grow up trillions upon trillions of E. coli cells, and all of them are going to start making this tomato chemical. You know, they're not going to look like tomatoes, and they're not going to smell like tomatoes, but they're going to make this one component of the tomato repertoire of chemicals that they make. So that's called recombinant DNA. When you take DNA from one thing and add another DNA into it, that's genetic engineering. So recombinant DNA. And often bacteria are the vehicles for these pieces of DNA. I'll give you another example. Insulin production. One of the advances in insulin production involved genetic engineering. They took from pigs DNA that would code for insulin. It turns out that pig insulin is very similar to human insulin. They took the pig insulin gene and incorporated that into E. coli DNA. All right, so let's say, let me draw a little picture to illustrate this. Let me, how do I do this? So let's say here's I was going to put in a slide here and draw on that, but here's an E. coli cell, okay, and this red in the center is its DNA. That's its chromosome. And what you can do is take that chromosome out of there and chemically cut a little chunk out of it, all right? Open it up. It's still active DNA, but it's got a little chunk out of it. And then take some DNA from another organism, say pig, that piece of DNA having the insulin gene in it, and splice that in there. Ooh. All right? There's our recombined DNA. And that little section with the insulin gene now can be expressed. What we can do is go ahead and place that back into an E. coli. I don't know why it made my blue. Now that E. coli cell will start to express that insulin gene. All right. And what's more, when that E. coli divides, it's going to make a copy of the insulin gene. And when those two cells divide, you're going to have 4 copies. And when those 4 cells divide, you're going to have 8 copies, and on and on and on. Now, it takes about 15 minutes or so for an E. coli cell to divide. You can maybe sit down and think this through. Just take a piece of paper and follow through. 1 cell dividing into 2 after 15 minutes, 2 into 4 after a half an hour. At the end of an hour, you'll have 16 cells. If you were to, say, allow that to continue unabated for, say, 48 hours, how many cells would you have? You're going to have a lot of cells. The exponential increase in the number of cells. All of them, all of those billions of cells making insulin. So at some point, you're going to have enough insulin that you could extract that and start to put it into vials and administer that to diabetics. That is genetic engineering. That's one of the valuable things that came from that. And there's a number of things that are produced in that very same way, even now. So yeah, recombinant DNA technology, making recombinant pieces of DNA, taking DNAs from 2 organisms and sticking them together, as a central part of genetic engineering. We now have a lot of genetically modified crops, by the way, and GMOs. You've heard about those. Those are newsworthy. Genetically modified organisms are organisms that have had their DNA recombined with DNAs of other organisms to give them desirable qualities, desirable characteristics. Maybe one of the things you want is a plant that doesn't get killed by herbicides. Only weeds will get killed by the herbicide. The crop that you're growing could be sprayed with the herbicide and won't kill it because it has been genetically engineered to withstand that. Or maybe you want a plant whose fruit doesn't ripen so fast that it rots quickly. You can genetically engineer plants to do that. So our GMOs are here to stay. You go to the produce section at the store, a lot of those plants are genetically engineered. They are GMOs. They are products of GMOs. That doesn't necessarily always involve using microbes, but I did want to make that point to you that that's genetic engineering. Then we've got bioremediation. I like to highlight things with red for some reason. Bioremediation. This is where you use microbes that sometimes have been genetically engineered to clean up the environment. So if you've had an oil spill, all that oil, sometimes millions of gallons, hundreds of thousands of gallons, fouling the environment, say shorelines or rivers or whatever. We now have bacteria that have been genetically engineered to literally eat that and break the material down into things that are less harmful. That's pretty amazing that we can do that. Also, heavy metals like lead and arsenic and cadmium and other things like that have been released from rocks, say at mine sites and so on, abandoned mine sites. Those are toxic places. We've got whole towns in the U.S. that had to essentially shut down because they were so toxic from all of the mine tailings that accumulated in East Oklahoma and parts of Kansas and elsewhere. Well, cleaning up those places, making them habitable again, might involve the use of microbes for bioremediation. So that's what that's about. All right, so I'm going to stop there. That's enough for our first lecture. I know I've waxed on for quite a while, but I'll put up several of these. I'll place them on eLearn. I'll put them under the course material section, but I'll also place them in the news section. So as they come up, they'll be right there. You'll get a little headline that says, hey, here's a new lecture number 2, lecture number 3, whatever, on whatever particular chapter we happen to be. That's a big enough snake for you to try to catch right now. You can handle that first lecture, and I'll just go ahead on and add to that as we go. All right, so we'll and oh, by the way, our semester this term is longer than it normally would be. Normally a 5-week semester. I think we've extended it out into a 10-week semester. So I'll have to look and see. I'm not even sure when the end date is, but we'll base our exams. We'll stagger them through the term so that they occur regularly through the term. So you won't be having an exam this first week, but for sure, likely an exam, an electronic exam available for the end of the second week. I'll give you the exact date and all of the details about that. And generally with electronic exams, I try to leave them up for a day or 2 or 3, at least a couple, 3 days to give you time in there to be able to take it, right? So not like you're going to have to be there front and center at a particular time to take the exam then. I'll give you a range of times. I will tell you that there will be a couple, at least, of our exams that will be administered by an outside organization known as HonorLock. So they have a way to monitor the exams, and it's just a good policy. I think it helps us to keep things on the up and up, so that the student that's registered for the course is the actual person taking the exam, and cut down on that sort of possibility. Not that anybody in this class would cheat, but we've got, that's a concern of ours. So anyway, more on that later. I'll go ahead and post this, and we'll just go on from there. All right, thanks.